Hello, everybody. Thank you very much for inviting me to talk here today. So I'm gonna switch gear a little bit from brain activity to just pure behavior. And I'm gonna tackle a relatively simple question, but it'll get a little more complicated as we go. So when we make saccades looking around in the world, we can actually move our eyes in all directions. We move our eyes horizontally, vertically, we can make oblique saccades. But it turns out that at least for humans, all these directions of saccades are not equally likely. We make a lot more saccades, as shown here in this cartoon depiction by arrows. We make more horizontal saccades. And then we make quite a bit of vertical saccades, but we make very few oblique saccades. So what we're gonna try to start to tackle today is kind of why this may be. And here is an actual data recording and summary of this effect. And we're gonna see quite a bit of these plots today, so I'm gonna try to describe it well today, right now. So here we have a plot that shows the frequencies of saccades for different directions. So the blue line shows for each direction how many saccades or what was the probability of a saccade in a given direction. And for this example here, which was recorded during free viewing of static images, we see that there are many saccades towards the left and the right. There are quite a few going up, a little less going down, and even less in oblique 45-degree approximately directions. So this is a very robust effect. It's seen in humans in multiple tasks, and I wonder why. So if we start thinking of possible hypotheses, I think the first one that usually comes to mind is just thinking about the symmetry of the system. So if we think of the eye muscles or the brain areas that control saccades, they have this horizontal symmetry in general, where pulling one eye to one side is controlled by one side of the brain and one muscle on one side, and pulling the eye towards the other side is symmetrically controlled in the other side. On the other hand, for vertical eye movements, things get a little more complicated. We have a pair of muscles that also produce torsion, and it takes more computation and more control to move the eye perfectly vertical or obliquely. So maybe horizontal saccades are just easier to program for the brain or somehow more energy efficient, so that's why we make more of them. But this gets very soon kind of disproved, or at least challenged, by a fact that has been shown by previous studies already, that if you simply tilt the visual scene that a subject is looking at, then the direction of saccades rotates with the image. So when the image is upright, most of the saccades the subjects make are horizontal, but when the image is tilted and the head is still upright, now oblique saccades become the most likely. So if it was really that much more costly, that shouldn't happen. You should still make saccades horizontally most of the time. But then we're gonna see that it's not really just the image itself. So for example, if we look at saccades that we make when we just try to fixate at a spot, as Jake showed, when you fixate at a spot, your eyes are still moving, and they'll produce small saccades, usually called microsaccades. So if we look at the direction of those microsaccades made during fixation, they show a very strong directional bias as well. So most of the microsaccades in humans tend to be horizontal. And this happens when people are looking at a simple fixation spot in the middle of a blank scene. So that would suggest that it's not really just about the visual scene, at least not the visual scene that you're looking at, but there may be something about the regularities on the visual images that we look at or the behaviors we usually have that produce this bias. Okay, so what are we gonna show today is a few experiments and analysis that try to at least tackle some questions to start to understand where this bias comes from. First, we are gonna show experiments where we are trying to really understand better what the reference frame of this bias is. So we are gonna try to decouple the head orientation in space, the orientation of the image in the world, and a little bit of the eye orientation of the head to see where this bias really stays anchored to. Then we look at how the bias is different maybe for different saccade sizes. And finally, we are going to try to analyze the images to try to predict what are the features in an image that may influence this saccade bias. And this is all work that has been done by a grad student in the lab, Stephanie Reeves, and she's a vision science graduate in Berkeley. So in the first experiment, we are using virtual reality. So we have this headset, the FOV, that is equipped with eye tracking internally. And we are going to show subjects either these fractal scenes that have the main characteristic that they are rotationally symmetric. So they don't have any cue for where upright is. And then they're also going to look at natural scenes that, of course, usually tell you where upright is. And then we are going to keep their head either upright or tilted. Here is the complete set of conditions in this experiment. As we see, there are 3 different head tilts. And then within each head tilt, we have 3 different image tilts. So we always have either the image aligned with the head or tilted 30 degrees more than the head or 30 degrees less than the head. And as I said before, the images could be the fractal images or the natural scenes. So first, we're going to focus on those fractal images. Now, it's a simple case where we have just a head tilt and an image that really doesn't have any tilt information. It's always the same. So in this scenario, we could think of 2 possible hypotheses. So when the head is upright, the 2 reference frame, either the world reference frame or the head reference frame, they are really the same. So with the 2 hypotheses, we get the same saccade distribution depicted here by this ellipse. But then when the head is tilted, we could get these 2 different results. The saccades could go with the head or they could stay with the world. So let's see what happens. So here we have the typical distribution for upright. And now subjects were just really previewing these fractal scenes. But then when the head is tilted, we see that the saccades rotate with the head. So they stay in a head reference frame. When the head is tilted to the right, the distributions shown here show a tilt to the right. And when this head is tilted to the left, the saccades tilt to the left. They go with the head. But if we start to look into the detail, it may seem that it's not exactly with the head. So we are going to do a further analysis. And this is how the data is going to be shown most often today, where we actually just look at how much this distribution deviates from a head reference. So now we've rotated the distribution. So the horizontal line represents the head orientation. And we are comparing in black, the distribution when the head is upright, and in blue or red, the distribution when the head is tilted. And when you do these plots, you start to getting a hint that there is a small rotation, but we can further quantify that. We use some cross-correlation analysis to measure how much you need to rotate one distribution to better match the other distribution. And we see that there is actually a small shift of these distributions where when the head is tilted to the left, the saccade directions rotate a little bit to the right. And when the head is tilted to the right, the saccades rotate a little bit to the left. And initially we try to quantify with just a summary index for each subject, how much their saccades remain in a head orientation reference frame or in a world orientation. And we see that, as we showed before, it's very close to the head, but not exactly right there. Now, okay, how is this? Good. So yeah, I was mentioning that there is this small deviation from a pure head reference frame. And this amount is actually consistent with the amount of eye movements that are produced usually with a head tilt. So when the head tilts, we get a torsional rotation of the eye, which is a rotation around the line of sight. And typically for a head tilt like here of 30 degrees, the eye is going to rotate around 4 or 5 degrees in the opposite direction. So this starts to suggest that maybe this bias is not in a pure head reference frame, but actually maybe in an eye reference frame. Okay. So next we are gonna focus on another set of conditions where the image is always horizontal respect to the world, but now the head may be tilted. Remember, this is still done in virtual reality, so it's not a completely natural condition, but it's the set of conditions that more mimic maybe the natural behavior, where you may be looking at the world when your head is tilted. And in this case, again, we find that the saccade directions deviate even more. So we don't get just this eye reference frame, but the saccade directions rotate to align themselves closer to where the horizon in the world is. But remember, these graphs here, now they are showing head orientation. So horizontal means aligned with the head. So when the head is tilted, this moves in the direction that would align it closer to the world. Okay, and finally, we have sort of the opposite condition where the head stays just upright. And now the image may be tilted. And this is just replicating previous results, but it shows also that the saccades rotate so they align with the image, but only partially. If we measure the angle that the saccades rotate, we see that even though the image was tilted 30 degrees, the saccades distribution rotate about 10 to 15 degrees. And we can represent again this here with this reference frame index, where 1 would be a perfect orientation with the image and 0 perfect orientation with the head. And for most subjects, we end up getting somewhere in between. So that's what the next experiment is going to start to tackle. We just purely here, we're going to use image tilt. The head is always going to be upright. And we're going to have 2 different behavioral conditions, one where they are freely viewing the image and one where they are fixating at the center of the screen where there is a small dot. And now the image can be tilted 30 degrees to the left or 30 degrees to the right. Now, overall, this is the result we get, and we've seen a few of these already. When the image tilts and subjects are free viewing, the saccade directions rotate. Now, surprisingly, potentially initially, is that when subjects are fixating in a dot and the image in the background is tilted, these microsaccades that are made during fixations don't change at all. So the distributions of these microsaccades remain exactly the same, no matter if there is an image in the background that is tilted or upright. Okay, and we can quantify this in the same way as shown before. So during free viewing, we get a big rotation, about 10 degrees, with a reference frame that we would think is closer to an egocentric, so a reference frame in the head, but it's still very affected by the image. On the other hand, for fixation, we get no effect, and the saccades are made in a purely egocentric head reference frame. Now, of course, this could about the task. In one case, they're fixating, they may be ignoring the background. So it may make sense that they are not affected by the background because they're just looking at the dot. And in the other case, they're actually previewing and engaging with the image. So then we did a further analysis where we just look at the free viewing data, but we group the saccades depending on their size. So we did 4 quartiles where we get the smallest saccades made during free viewing, less than 1 degree. Then we have other groups from 1 to 2, more or less, 2 to 4, and bigger than 4. What we clearly see is that there is a pattern that changes. So for the big saccades, we get a very strong effect of the tilt of the image. But for the small saccades, we get almost no rotation. And again, we can quantify with this reference frame index, where 0 means aligned with the head and 1 means aligned with the image. And small saccades remain aligned with the head, but big saccades align more and more with the image. Okay, so after doing this, when we look more closely at the data, we find that not all images are the same. If we were to show what these effects are for different images, we'll find images that have a very big effect, meaning they pull the saccades to be oriented with that image when it's tilted, and other images that don't seem to have the same effect. So what we are trying is to find what are the features, the characteristics of those images that could predict which images affect the saccades and which ones don't. So in the first option that we thought of, we are studying the saliency of an image. So this is something that has been done a lot in the field of eye movements, where you extract what are the most salient features of an image by contrast, orientation, etc. And we can build a saliency map, as shown here, that essentially would predict the positions in the image where it's more likely to fixate. So now you could end up with some images that have some structure on this saliency, and that the structure would potentially induce a bias. So if you have, like here, only 2 very salient targets, you could predict that the subject is going to be looking between those targets a lot. So you're going to get more saccades in particular directions. While on the other hand, you may have other images where the saliency map is more uniform. So it would not predict a lot of bias in the directions, just purely caused by this structure in the saliency map. As a second option, we are going to look at the spatial frequency distribution, similar to, again, what Jake was doing. But in this case, we're going to focus at the power of the spectrum in different orientations. So here I have, again, 2 examples. One where there is a very strong orientation signal, in this case, probably more in the low frequency, but also in the high, where you have a very distinctive biased distribution of power in other images. You may have a more uniform power in all directions. And finally, the third option of feature we're looking at is maybe the harder to analyze, but it's the more cognitive, the cues about where gravity is or where the floor is. So this cannot be directly extracted with low-level features. So we are using a deep learning network trained with actual images of known orientation. And that network can tell us what the orientation of that image is, of any image, and how certain the network is of that orientation. So then we can end up with images that clearly tells us where upright is, and other images that may not have so much. So with these 3 options, we are gonna do essentially the same analysis. With all of them, we get a metric that tells us how the saliency is strongly biased, how the frequencies are strongly biased, or how strong the deep neural network can tell us where upright is. And we can correlate that with the strength of the effect on rotating the saccades that I shown before. And this is the summary of the result. And what we can see, at least preliminary, because this is still a small set of images, is that the spatial frequency, either low or high, seem to be the strongest contributor to this effect. So to summarize, today we've shown that saccade generation is not uniform in all directions. We humans make saccades especially horizontal, and this may not be true for other animal species, which is another interesting line of approach to this problem. We have this big bias towards horizontal directions and less so to vertical. And this bias is not really fixed, neither to the head or the world. It's probably initially biased towards the head, but it can be affected by the image content. And this is especially true for larger saccades. Larger saccades seem to take more information about this tilt of the image and reorient themselves with the image, while the small saccades are more tightly tied to the head. And then the spatial frequency content and how it is directionally biased seems to be the best predictor for now about the effect of different images on this saccade tilt. So thank you, everybody. And I want to thank also the people in my lab, especially Stephanie Reeves and Raul Rodriguez, which contributed to this work, and the funding agencies. And that's my eye doing a little bit of torsion. Okay, thank you.
So that was really interesting, basically telling us that saccade directions are modulated by the information in the image. So saccades are seeking the information. Do you guys have any questions? Laura, is that a hand?
Yeah, thank you very much. Nice talk and very nice project. Do you think that is related somehow to listings plane?
So the initial data that I showed with the small effect, that could be, but the effect of the image is so much bigger than anything that you could really predict with the listings plane.
Thinking rather of the microsaccades, which are less sensitive to the image.
Yeah, no, certainly. But still, I don't think that all saccades are generated by the same circuits as far as we know right now. So I don't think enforcing the restrictions of listings plane would necessarily predict why the small ones versus the big one would be differently affected by the image.
Thank you.
Thank you.
Thank you, Jorge, for your lovely talk. Being somebody who studies development, you know where I'm going to go. and so I can imagine the spatial frequency piece being quite reflex. And then I can imagine the world and the task and the free viewing being more learned.
Mm-hmm.
Are you willing to speculate about any of that?
Not too much, but I think
That's
there are
fair.
some studies that have shown, so people have looked at the horizontal bias across ages. There may be one study that I know of, and it seems to become more and more biased with age. So it starts more with saccades made in all directions, and with age, the distribution becomes tighter and tighter horizontally. But I don't know the effects of tilting the image, if that would independently change with age or not. No idea. Thank you.
Hi, you said that spatial frequency influences. I want to know what spatial frequencies do what.
Yeah, so right now we essentially we tried to look at different spatial frequency bands, but we didn't find a different effect. So we found the same result for low or high right now. So if we just look at how each band of frequencies is biased in directions, we see the same effect.
Hello.
Hello.
Very cool talk.
Thanks.
So when you say it goes with the head, I mean, there's two ways you can think of that, right? It can go with the eye line, or it can go with the vestibular signal of the head,
Mhm.
right? So if you were to have a situation where the tilt does not go with gravity, let's say a person's lying down, right, and they're doing the same head tilt, how would you predict that the behavior would change?
Yes, good question. So, but I think since we see that still stay with the head, so it's not aligned with gravity, I would expect to still go with the head mostly when even is the eye line, yeah, or the roll direction of the head. Yeah, thank you.
My question is, how do people scan an entire scene if they're primarily only using horizontal saccades? Are these coming back to the same place and then going up, or are these actually two unique targets?
That's a good point. I forgot to clarify. So in those directions, we ignore size. When we say there are more saccades in one direction, they could be more small ones where you have more big ones in the other direction. So I think in general, we'll cover, you can cover the entire field, but you're going to switch more horizontally. But you're going, and still, those saccades are not perfectly horizontal. They still have an oblique component, so you can zigzag around the image. But yep, it's interesting. It doesn't seem the most efficient thing to do it to extract information.
Okay, so we have a longer discussion section at the end of every session. So if you have more questions, let's reserve it for then. Thank you, Jorge.